Hey, I'm Josh.
Hey, I'm Noah.
Today we're going to talk about genetics.
Genetics is the study of heredity and the variation of inherited characteristics. This is meiosis. Meiosis is one of the main parts of genetics. Meiosis is where your sex cells split to cause genetic variation from your mother and your father. There are 9 steps. Interphase is the dormant step, or things are happening. There's 3 phases in interphase. There's G1, S, and G2. G1 is growth. S is where it copies your DNA, and G2 is where it prepares to split. Prophase 1 is where the nucleus starts disintegrating and the spindle fibers start to form, and crossing over begins. Metaphase 1 is where the spindle fibers attach to your chromosomes, and crossing over is still happening. Crossing over is when the chromosomes cross over to create genetic variation. Anaphase 1 is where the spindle fibers pull the chromosomes apart to separate ends of the cell. In telophase 1, the chromosomes that were pulled to separate parts of the cell are now in their own cell, forming cell, and the nucleus starts to reform. In prophase 2, the nucleus disintegrates again, and crossing over isn't happening anymore. In metaphase 2, the chromosomes line up in the center of the cell, and the spindle fibers attach to them. In anaphase 2, the chromosomes are pulled apart to separate ends of the cell, and then in telophase 2, the chromosomes start to have their own cell, and the cell starts reforming, and the nucleus starts reforming. And then at the end of meiosis, you have 4 daughter cells that are genetically different from each other.
Now we're going to talk to you about how we have different features with genetics. Rutger Mendel was a scientist that created the 3 laws. Law of segregation, which is 2 alleles for each trait, separates during meiosis. Law of independent assortment, random distribution of alleles occur during gain functions. And the law of dominance, the dominant allele shows. Reginald Punnett developed what is now called the Punnett square. He developed a monohybrid cross, which is a cross between 1 gene. He also created a dihybrid cross, which is a cross between 2 genes. And to find out the genotypes for the dihybrid cross, you have to use the FOIL method, which is first, outer, inner, and last.
This is a pedigree.
A pedigree is a diagram that shows the patterns of inheritance of a trait.
A pedigree has different symbols for if you have a disease or if you're a carrier of the disease. If it's a circle, it's a female. If it's a square, it's a male. If it's half filled in, you're a carrier. If it's fully filled in, you have the disease or trait. If, for example, if your parents have a disease, they could pass it down to you and you could have the disease, or you could just be a carrier for the disease, which would mean you're half filled in.
If your parents have the disease, you also have a chance of not getting the disease or being a carrier. We have non-Mendelian genetics. This means that they don't follow Mendel's 3 laws.
There are 3 pieces to non-Mendelian genetics. Number 1 is incomplete dominance. Incomplete dominance is when the recessive allele isn't fully hidden from the dominant allele.
Next is co-dominance. This means that both alleles are fully expressed. An example of this would be when you have a white flower and a red flower and the offspring becomes pink.
Third is multiple alleles. Multiple alleles is when there's multiple alleles in one set of organisms.